Today we'll be talking about a distinct series of brain areas that are specifically used for learned vocalizations in a group of species that you maybe don't spend a whole lot of time thinking about. We're going to be talking about the song control system, which we find in the brains of songbirds. So why don't we get to it? Let's talk a little bit first about vocal communication. We're going to talk about the role of song in songbirds. We're going to talk about the brain circuits that are involved. And then we're going to finish up discussing how some of these similarities of how this circuit works correlates to what we understand to be true in mammals and in humans. Communication can take many forms. We talked about different forms of communication in the last lecture. We obviously covered speech in great detail. We also mentioned writing. And of course, there's sign language as well, so we discussed all three of those. There's also body language. There's a lot that the face can communicate, as well as the rest of the body. This sweet boy is trying to communicate that he wants to play, ready to play. He's looking at us intently with eyes wide open and wagging his tail. People and animals can use body language to communicate with each other. This boy right here is not too happy. I'm imagining this guy stole his bone, and now he's very upset. You can see expressing growling, showing the teeth, very aggressive behaviors coming out of this boy right here. And this sweet boy right here is showing submissiveness, the anxiety. His ears are down below, and he's pretty, cautious. And he's saying, okay, fine, you win, take your bone. You can find all kinds of communication within animals, and the more complex the social structure within animals, we find more complex languages. This is true for honeybees, in fact. Their language, in fact, has very specific meaning. They do this waggle dance. So this is a bee that's making this little wiggle waggle and then does a little curve. There's actually specific meaning to the wiggle waggle here. So what they're doing here is that they're communicating to the other bees that nectar is located in a particular location. And the way that they are doing this is that the angle in which the wiggle is happening on the comb is relative to where the position of the sun is in the sky relative to where the hive is. And then how long the wiggle waggle is directly proportional to how far away the nectar is. So it's giving direction and distance information to the other bees so that they can go and find the honey source as well. And of course, there's vocalizations. Lots of animals vocalize. Dogs, of course, they vocalize. They howl, they bark, they growl, they whine. There's a lot of specific meanings behind their vocalizations. This is true for many, many animals. But the thing is, most animals don't actually learn their vocalizations. Dogs don't learn their vocalizations. They're just able to do it. We're going to talk about learned vocalizations. So let's talk about the different animals that do learn their vocalizations. There aren't too many categories of animals that learn their vocalizations. We know that there are learned vocalizations from bats. Bats learn their vocalizations. We know that whales and dolphins, they have complex vocalizations that they can generate. And then there are different birds, groups of birds that we know learn their vocalizations. Parrots famously learn their vocalizations, many different species of parrots, and they all seem to have a very complex system of learned vocalizations. Hummingbirds do as well, as do songbirds. And of course, then there are humans. Humans learn their vocalizations, and we talked about that in the last lecture. So when we're trying to understand how the brain generates learned vocalizations, learning an auditory template and generating a motor output, we don't actually have a lot of animals to choose from. You can do some work with bats, but they're not the easiest thing to keep in the lab. Whales and dolphins are definitely not easy to keep in the lab. So that leaves us with only a few choices, and that's where songbirds have become. We talked about the sonogram in the last lecture, about how we can have a visual representation of what a sound looks like. This has been heavily used, of course, in studying singing behavior in songbirds. And again, a sonogram is used to visualize sound. This is an example of a bird that's been widely used in songbird research and singing behavior research. It's called the white-crowned sparrow. You could find these guys locally as well. They tend to migrate through Virginia, but in the winter months, you're going to find them in sort of brushy areas. I see them often in the brushy areas that are sort of near the tennis courts along the bike path that goes down to the Corporate Research Center. So you can see that their vocalizations these guys at least don't have a whole lot of harmonic stacks going on. They instead have some specific whistles, and then they make a warble, and then they have buzzes. When you think about it, if you know anything about songbirds, they all have different kinds of songs that they can sing. And that's definitely true with white-crowned sparrows. Again, just to illustrate what a sonogram is made of, we have frequency that is represented on the y-axis here. We have amplitude represented by how intense the mark is, and then time is on the x-axis. So this bird would be singing a whistle, and then a warble, and then a buzz, buzz, buzz. Sometimes this is a trill. Okay, so those are white-crowns. We're going to talk about the Australian zebrafinch, which is really the lab rat of the songbird world. So zebrafinches come from Australia. They are widely dispersed across Australia, and they live in some pretty rough habitats. And that's actually made them kind of ideal as model species to work with in the lab. Because of these difficult habitats they live in, they're opportunistic breeders. And what that means is if they have resources that are available, water and food, then they will breed, and they will breed a lot. The interior of Australia is a very tough place to live, and they only can breed when they can. But under captivity, you can provide as much food and water as you want, and then these guys are just going to breed all the time. So that makes them really great to work with the lab. Most songbirds will breed only once a year, usually in the spring, and so that's typically how that is done. They are sexually dimorphic. You have males and you have females. They're very obvious in their coloration. They are domesticated, so they have been a domesticated species for over 100 years, used as pets. Maybe some of you have had zebrafinches at home. And, of course, they're widely used for research. So this is what their sonogram looks like. It is a series. So zebrafinches, they don't have quite as pretty song as other songbirds. They tend to have a little bit more of a buzzy sound, but it's very specific, and it's learned, and it's pretty easy to quantify. You can see that actually it's somewhat similar to human speech in that it has a lot of harmonic stacks. So they're generating all kinds of formants that have different emphasis at different places. So they will make a series of introductory notes. That's what I is here. And then we indicate the different notes with a letter underneath. And you can see that this bird is making a series of 5 notes, A, B, C, D, E, and then it repeats it, A, B, C, D, E, A, B, C, D, E, A, B, C, D, E. It does it over and over again. The subdivision of the repeated notes is called a motif. So this bird, in this particular song, sang 4 motifs in a row with a couple of introductory notes. So let's see if we can play the song. Well, in the PowerPoint, you'll be able to play this song. So you'll be able to click on this little thing, and then you can hear the song. Unfortunately, I can't include it in the video. So birdsong actually has some very interesting parallels with speech, but it's not language. So both humans and birds, this is a little juvenile zebrafinch here. So they have sort of a darker plumage. They don't have the bright coloration here, but this is a male. They have an acquisition of a vocal repertoire by imitation of sounds. Both humans and songbirds need to hear some kind of vocal output, usually from parents, and then they will repeat and copy those sounds and then generate it as adults or as juveniles. But that's where a lot of the similarities break down. Human language obviously has symbolic representation and syntax. When we make our series of phonemes, there's actual meaning behind them. I say the word bicycle. You can envision what a bicycle is. It has symbolic representation. Also, the order in which is really important. Now, birdsong also has syntax to some degree. You saw the zebrafinch song before, that there's a specific order, A, B, C, D, E. So there's an order in which they will make their songs. You can't just randomize them, and in fact, birds will have a hard time recognizing randomized order of notes. The notes generally will have to appear in a particular order. So there's some sort of syntax, but there's no grammar, right? There's not any sort of underlying meaning. It's not like the note A means something specific. Instead, it is being used primarily for courtship and territorial behavior. That is the main reason why they sing. They sing to attract mates and establish territories, which we'll talk about in more detail in the next couple of slides. Now, with humans, of course, we use language for courtship and territorial behavior, but we use it for a lot more than that. Because our language, we have symbolic representation. You can have infinite meanings. So you can have, in the human English language has something like 50,000 words with only 44 phonemes. There's 44 phonemes, and we can make thousands of different words. Birdsong undergoes a series of stages of development that are actually quite similar to the way language is learned in humans. So we're going to walk through these different stages. The first phase is the sensory phase. So this begins right at the beginning. And if we're talking about seasonally breeding songbirds, it's typically within the spring. That's when birds are hatched and when they're born. During the sensory phase, a juvenile songbird must hear a conspecific song in order to make that song as an adult. The word conspecific means a song from a species, a member of that same species. So a white-crowned sparrow is going to learn the song of a white-crowned and not, say, of a song sparrow or a chipping sparrow or a Swanson sparrow. They're going to learn white-crowned sparrow song. And that's true for most species. There are some species that will copy the songs of other songbirds. This phase is dependent upon auditory feedback. So here's the little baby chick, and it's listening to its dad sing. It's usually males that sing. So this male is singing away because it's spring and it's still breeding season, and he's going to establish his territory. And this little bird is listening and trying to memorize and is memorizing that template. Again, dependent upon auditory feedback. So if you deafen a bird during this phase, they will not. The next stage is known as the sensory motor phase. During this phase, the juvenile songbird starts to practice his learned song. It starts off with what's called subsong, and it's really quite variable, not very good, and it sounds an awful lot like baby babble. Near the end of this phase, the bird starts to sing what's called plastic song. The song starts to resemble what the learned song was, so it's pretty close in what it looks like to the male tutor that it learned from, usually the father again. But it's still quite variable. So from rendition to rendition, there'll be some changes in the notes, and it's not actually a perfect match. This phase is also still dependent upon auditory feedback. If you deafen birds during this phase, during the sensory motor phase, they fail to make a good, adequate song as an adult. Once a bird reaches sexual maturation, the song is crystallized. And in most seasonally breeding songbirds, it's typically after just 1 year. In the next breeding season, they are now crystallized. So the song is now mature, it's stable, and it's very, very similar to his tutor song. And once crystallized, the song is largely no longer dependent upon auditory feedback. So if you deafen birds, even at this stage, you'll get subtle and small and slow changes in the song over time, but they can still mostly make the same song for a long period of time. So we're going to talk about some of the examples of what this looks like. This is from some classic experiments done in the late 50s and early 60s. So these are sonograms of different song sparrows. This bird was put into a soundproof chamber and was played, the song above, this song right here, was played to this bird. So this bird was in a soundproof chamber, didn't even have a tutor to look at or interact with. And these are from song sparrows, where they do a pretty close match of the songs that they're actually learning. And you can see from just looking at the sonogram, this is why sonograms are so helpful, that it's pretty close. It's got like these 4 notes right here, and then it has a very similar B note, and then it has this sort of buzz noise that it makes, so that's C. And then it has 3, not 4, but it has 3 of these warbles, and then copies this end component here. So even though this bird is isolated, it's getting this song as its template to learn, and it does a pretty good job. It's nearly identical. This is a pretty good example that these songs are learned. This is an example of a bird that was isolated but never heard conspecific song. It has some basic elements of what you would consider to be song sparrow song, but it's these notes, if you could just imagine, they're sort of warbly, there's some structure, but it's not very typical. If we were able to play this song, it would sound like total garbage. It would kind of sound like song sparrow song, but it would be really weird sounding. It wouldn't have these precise kind of cuts offs and notes and nearly as melodic. This is what it looks like in a bird who is deafened at a very young age. So from this, you can just tell that this is really variable and really scratchy. There's some elements of like structure, like notes that are being sung, but there's really no commonality across the different notes. And instead, it's really variable and really strange. You can just look at the sonograms and tell that there's these huge differences. So this is an example of showing like what happens when these birds are deafened. Now, the other interesting thing about songbirds is that there's over 4,500 different species of just songbirds. Now, to put that into context, there are nearly 4,300 different species of mammals, all the whales, the rats, the mice, cows, everything, giraffes, humans, primates. There are more different kinds of songbirds than just mammals. And you can just think about how variable mammals are and the kinds of vocalizations they make. So it's not surprising that you can find all kinds of variability across songbirds. So there's a lot of variability. This is just 3 examples. And we've already talked about sparrows and zebra finches. They are examples of what are called closed-ended learners. So they learn during the sensory phase, they practice during the sensory motor phase, and then the song is quite crystallized. Because zebra finches have this opportunistic breeding system, and one reason why they've become a common lab animal is because they have crystallized song mostly by around day 90. They can actually reach sexual maturation after just 90 days, whereas for other seasonally breeding songbird species, it takes at least a year, so at least 365 days. So this makes them an ideal animal model to work with. But once the song is crystallized, they don't learn new songs past that critical period. But there are other examples of birds that actually are open-ended, and they can learn new songs as a sexually mature adult. So they go through the initial sensory and sensory motor phase, then it's crystallized. And then in the next autumn, they go through another sensory and sensory motor phase where they may incorporate new song elements into their repertoire, maybe even new songs, such as like a mockingbird, which can sing more than 100 songs, 100 different kinds of songs, copying songs of other different species and even machine noises in the environment. Canaries, everyone knows a canary. This is a pet bird. These are the classic example of an open-ended learner, and they've been studied for their ability to do open-ended learning. So as I mentioned, birdsong is used to establish territory. So this is going to be a little graphic that explains this. So these are pictures of different song sparrows, and they have their sort of specific songs. There'll be a lot that's in common because they're all living in the same territory, and their songs are going to have a very kind of specific dialect. In fact, just very similar to the dialects that we see in humans. There's a southern dialect, a northeast dialect, a New York dialect, a Chicago dialect. Birds will also have dialects as well. But birds will also learn that the different songs of the different birds are for that specific individual. So let's say this is Bob here, bird B, and he hears Adam is over here to his left and that Charles is over here to his right. And if he hears songs from Adam over here, he doesn't even have to see Adam, but he knows Adam's songs. He's learned that Adam is over here to his left. He's going to be okay. Like they've already worked out that this is Bob's territory and this is Adam's territory. But if you heard Adam's song over here where Charles is, he'd be like, wait a minute, Adam is supposed to be over here. Maybe that's not Adam. Maybe that's not Charles. I should go and investigate and maybe beat that bird up. So the way we know that the territories can be a problem is that, let's say Doug comes in here and Bob doesn't recognize Doug's songs, and he's going to go over and investigate and likely beat up Doug. Say, hey, you know what? That's not your territory. Get out of there. This can be tested in under laboratory environments. And there's a lab in our department, in the School of Neuroscience, Kendra Sewell's lab, where she does very similar experiments just like this, where she will do what's called a simulated territorial intrusion. This involves placing a male decoy in the middle of a focal bird's territory. Let's say we're studying Bob. We'll bring in a cage that has, say, Doug in here, and then we'll start playing a conspecific song. So we'll start playing the song of song sparrows that are different than, say, the songs that are there. Then you can watch the behavior and see how they react. And then this is a way of establishing how territorial a bird is. What is the territory itself? Like if I move this farther away, what is the likelihood of the bird coming in and reacting? And you can map out the territory using this procedure. So it's used to establish territories, but it's also used to attract females. So there are a bunch of different experiments that have been done to test the degree to which song is used to attract females. This is a famous experiment done several decades ago now using pied flycatchers. So they had female pied flycatchers in a caged area, and they had 2 nest boxes that were available to them. These females, they didn't have any males there, and instead there was a speaker above one nest box that was playing pied flycatcher song, and the other nest box had no song being played whatsoever. And out of 10 of the female flycatchers that they examined, 9 of them established nesting behavior in the nest box that was playing pied flycatcher's song. So they were attracted to that nest because there was a male there, or just the male song, not even an actual male. And then that's where that they preferred that nest versus just one that had preferred the nest with no song. This is one classic example of showing that females prefer birdsong, and it's used to attract females. In some species, birds with bigger song repertoires actually have more babies. So this shows that it can actually be a sign of being a successful, strong male. So again, here's another picture of a song sparrow. Song sparrows, they actually will have different size repertoires. They don't have huge repertoires like a mockingbird. Their repertoire size can be as little as like 3 to 4 songs, or as big as like 12 songs or so. So it's a pretty big range. This is 3 times the size of these guys. What we have seen, and this is data clearly showing this, is that with different size repertoires, you see that there's a direct correlation with larger repertoires leading to more overall lifetime reproductive success. And we would define lifetime reproductive success as having successfully mated with a female, hatching eggs and raising those eggs, and then having those eggs, the chicks, reach their adulthood stage as well. You can measure these things and monitor them over time, and you can see that males with larger repertoires have overall lifetime reproductive success that's higher than those song sparrows that have smaller repertoires. So it's clearly something that's sexually selected, and that there's been pressure for birds to develop larger repertoires, and it's a sign of overall strength and virility. Now we're going to talk about some of the physiological mechanisms that underlie the production of song and songbirds. Birds do not use their larynx to generate sounds. They do have a larynx. It's located in the same place where the human larynx is, but they don't have vocal folds up there. Instead, they have what is called a syrnix. The syrnix is a series of muscles that are at the junction of where the trachea splits off to the bronchi that go to the different lungs. And because of this, they essentially have 2 sets of vocal cords. And this has been one of the reasons why birds have such varied ability in producing different kinds of sounds that humans can't do. So it's fundamentally different than the larynx in humans. And we'll talk a little bit about it. So here's the syrinx here, located way down below at the junction of the trachea where it meets the bronchi. So I'm going to tell you about an experiment that I was fortunate enough to be a part of, where we used what was micro CT scanning. So we used a CT scanner, but this was a very special scanner that allowed us to generate a 3D model of anatomical structures. So you don't have to memorize these details, but I just want to explain what micro CT scanning is Essentially, you use X-rays, and you're passing these X-rays through a specimen, say a bird's syrnix. And so you're passing these X-rays through, and it goes through and is collected onto a detector. And then you rotate the specimen, and then you take another X-ray and you do it, and then you have another image where it's slightly rotated. And you do this over and over and over again until the specimen makes 360 degrees rotation. And then you have all these different images of the 3D structure that's been projected at a 2D system. But because then all this information is fed into a computer where they can recreate a 3D representation of what that looks like. So we did this with songbirds, zebra finches, and we were also doing this to look at the syrnix. So this is actually like a 3D model of an entire zebra finch skeleton, because these are X-rays, so we can observe just the bones. And in fact, it turns out that there were like bones that are within the trachea. Each of the rings of the trachea has some ossification. We were able to see the syrnix way down here. We also did some MRI scanning as well, where we could, with the MRI, you can see the soft tissues such as the muscles of the syrnix. So this would be a muscle of the syrnix. And there's the trachea here. We also could trace out heart and lungs, all right there at the base of the trachea. We went into further detail where we were looking at the overall skeleton of the syrnix. And so this is the trachea here. These are the bronchi, and here's the branching here. And they have a very complex skeleton right at the syrnix, where they have little attachment points for the muscles that would connect here to here, so that they can control the vocal folds that would be located on one side or the other side. Zebra finches are sexually dimorphic, and it turns out that the skeleton of the syrnix in males is much bigger and more robust than it is in females. They have a similar overall anatomy, but it's just much more robust in males. We were also able to then trace out the connections of the different muscles, and we identified all the different muscle groups that are involved. We identified at least 8 different muscle groups on either side of the syrnix. So a total of 16 muscles to control these vocal folds. And we're talking about something that's a centimeter in length. This entire thing is about a centimeter in length and maybe 800 micrometers across. So it's a very small structure, but it was, it's an extraordinarily complex system. And so you have all these muscles, of course. So that's the key to this, is that this is a motor controlled behavior, and all these muscles need to be controlled by the nervous system. So we're going to talk a little bit about that. So the muscles are going to contract in a particular way, in a very particular pattern, in order to generate these sounds. And it's these muscles that does the job. Obviously, the muscles need to be controlled by the brain. So we're going to talk about songbird brains. This is where we're going to get a little bit of comparative neuroanatomy. So up to this point, we have almost entirely focused on just mammalian brains, but there are a lot more animals than just mammals. And there are some important differences, but also a lot of similarities. Now, one thing about non-mammalian brains, like the songbird, is that they don't have a laminar cortex. So this is an example of a cross-section of a human brain. And we know that in this area, this is where we're going to find all the cell bodies that make up the cerebral cortex. And that this would be the white matter down below, where you're not going to have a whole lot of cell bodies. We all have learned about this 6-layer system at the top of the cortex. Birds don't have that laminar structure. Instead, they have a very thick section at the top of their brain, which is akin to the cerebral cortex. We refer to this as pallium, and this is an old Latin term that it's kind of dependent upon the development, but in birds and the adults, we refer to this as pallium. And we can also refer to the same section of the cerebral cortex in mammals as also being pallium. It's pallial in nature. So bird brains are very small compared to the human brain, but they have a relatively high brain-to-body ratio. So this is another way of sort of measuring degree of intelligence. Animals that are more complex in their behaviors, especially social animals, animals that do a lot of complex learning, they're going to have a bigger brain relative to their overall body. Birds, especially songbirds, will have relatively big brains compared to their overall body size. Humans have the biggest brain-to-body ratio. Birds are more along the size of chimpanzees or dogs, more like chimpanzees actually. They're actually quite high in their overall brain-to-body ratio. So they have all the same basic brain structures, essentially in the same places. They have a cerebellum, they have a thalamus, they have a midbrain, optic tectum, which is the superior colliculus. But one big difference is that they don't have this layered cortex. All right. Within the bird pallium, they have their brain structure, instead of it being laminar, is that they have what are called brain nuclei. The singular form of that is a brain nucleus. So when we say the word brain nucleus, we're not referring to the nucleus of, say, a cell body where all the DNA is. Instead, a brain nucleus is a collection of neurons that are distinct in anatomy, where they sort of stand out. If you cut through a slice of a brain, you'll see this distinct group. And they are generally involved in the same function, in that maybe they're projected to say a different brain area, but all of those neurons in that area, or at least a subdivision of them, will project to a similar brain area to control some sort of function. Mammalian brains have brain nuclei as well. You can think of the globus pallidus as brain nuclei, the striatum as a brain nucleus. The cortex isn't really nuclear in its structure, though, right? Because it's this big long sheet, and it's difficult to distinguish one area from another just based on what the neurons look like. But the bird pallium is filled with these kinds of brain nuclei. How do we go about looking at what a brain nucleus looks like? Well, these brains are relatively big. A zebrafinch brain is about the size of your thumbnail. Like if you were to just look at that top part of your thumb, that's about the size of a zebrafinch brain. They're relatively big. You can't just look through the whole thing and just see, oh, those are all the brain nuclei. Instead, what we do, and this is true for if you're dealing with mice brains, rat brains, a bird brain is about the same size as a mouse brain. You'll have to put it onto some kind of device like a cryostat or a microtome. Both of these things have really sharp knives that you can then slice the brain like a meat slicer at the deli. And so you're going to get a series of slices, and if you keep track of those slices, you can track when a brain nucleus starts to appear. So let's say we're slicing sagittally, right? So this would be the pallium here. Here's the midbrain. Here's the spinal cord. And as we're slicing through, and we keep track of slice A, B, C, D, we can start to see a brain nucleus start to appear. We see some distinct neurons, and then it gets bigger and bigger, bigger and bigger, bigger, until we get to the largest cross-section. It may change its size and shape. It's going to get bigger and then smaller usually, because it is just kind of like usually ovoid in shape. But it might be more round at one side and then a little bit more ovoid and then on the other side. So the nucleus can change its shape. It's not going to be like a perfect football or an egg or something like that. But it's going to get bigger and smaller until we don't see it again. And we can track the size of this by tracing what the size is of the nucleus at one side and then going to the next one and measuring how big this is, measuring how big this is. And if we know the distance between these two sections, 40 microns, 80 microns, 120 microns, we can calculate the volume using that information. This is a picture of one bird brain nucleus, and you can see that these things are actually quite distinct, that they can stand out in the tissue. And so this is a Nisselstein section. These are all neurons and some glia, but it's very, very distinct and it stands out in the tissue. So when you're doing this, you're like, oh, okay, we see all these brain nuclei. What does this mean? Well, over the time, we have figured out that these different brain nuclei through the bird brain are play important roles. And we have worked out the pathways from the auditory inputs. So we've got the ear cells here, the hair cells in the ear, going through the cochlear spiral ganglion to the cochlear nucleus, up through the different nuclei within the hindbrain, the thalamus, up to primary auditory cortex, which is L2 in birds. And we can trace all of these things. So we know the interconnectivity of these things and that there are specific auditory pathways. So we know the connections, just like we do in mammals, the connections all the way from the ears up into the cortical-like structures of the brain. But then we also have the vocal pathways, and we have worked this out as well, and we know the connections from that auditory pathway. So this is part of the auditory pathway feeding into the vocal pathways that then control the muscles of the syrnix and the respiratory motor muscles that control the lungs so that you can force air through the lungs at particular times. So we've traced all the way from hair cells all the way out to motor units. You do not need to memorize all of these circuits, okay? I'm just showing you as an example that these circuits involve sensory pathways, motor pathways, and they're relatively complex. I'm going to show you a simplified schematic of what this looks like. But first, we're going to talk about how we know that brain nuclei are connected to each other. How do we know a brain area is connected to another area? When you're just slicing through the brain and you do the stain for the cell body, you don't see the axons necessarily. You'd have to do some special staining for that. We can figure this out by doing what's called retrograde tracing. So with retrograde tracing, what we do is that we can inject a dye into a brain area. So this is in a live intact animal. The animal is like a bird, for instance. We put it under anesthesia. We make a little hole in the skull, and we place a glass pipette down that's filled with this tracer, and it fills up a brain area. We target it to a specific brain area. Sometimes, usually this is, especially nowadays, it's usually a fluorescent dye that is used, but sometimes you can use a colored dye, so it literally is like a dye. These dyes are taken up by the axon terminals, and then they're transported up to the cell bodies of other neurons. So over time, you give it a few days, the axons terminals that are connected to this nucleus, from this nucleus, the neurons that are projected here, will have taken up the dye, and then those cells will now have the dye in them. And then here's an example of what this looks like. So you can see even the axons are sort of labeled in here, but the cell bodies are definitely labeled. So in this particular brain section, this is showing a cross-section of a brain nucleus that is filled with neurons that are projections, and the injection was made to some downstream nucleus that was receiving axon input from here. And so this is retrograde tracing, and this is one way that we can find out that brain areas are connected to other brain areas. So this has been done through the song control system. When we're slicing through the songbird brains, we see all these different distinct nuclei. And it turns out many of these distinct nuclei are specifically related to song. So we're going to talk about how we know that. This is a simplified schematic of the song control system. I'm going to talk about the two main pathways within the song control system. The first is the descending motor pathway, and that's in green. This includes HVC, which projects to RA, which then projects to the 12th cranial nerve, which is the hypoglossal nucleus. The 12th cranial nerve, this is N12 TS. TS means tracheosyringeal portion of the 12th nucleus. But it's the 12th nerve, N12 TS. This has a bunch of neurons in here that project to the muscles that innervate the syrnix. So what would be the neurotransmitter of those motor units when it's synapsing onto the neuromuscular junction of the syrnix? I'll leave you to answer that. But these are motor units, right? So it's pretty clear. This is like a premotor area feeding to this motor area, which then projects to, like this is essentially motor cortex. This is part of pallium, but then it projects to this brainstem nucleus, which innervates the muscles of the syrinx. So we know that these are the motor areas. So we're just focusing on this green part here. HVC, RA, area X. HVC, RA, and the 12th nucleus. I'm going to mention as an aside that the song control system is typically larger in males than it is in females. This is especially true of zebra finches. So here's an example of a male RA, and you can see it's pretty distinct here. And this is a female RA, really tiny, really kind of shriveled up. Males and generally are the ones that sing. They're using it to attract females, and this is distinct in the brain. When we talk about sex differences in the brain, we're going to talk about this in a little bit more detail, but I just want to mention as an aside that mostly what we're talking about here are males. There's some very interesting underlying endocrinology that's also related to this, which you'll be learning about in the laboratory portion of this class. And then we will talk about it when we talk about sex-specific differences in the brain. So how do we know that particular brain areas, such as the song system nuclei, are involved in particular behaviors? we can use what are called lesions. The way this is done is that you lower, say, an electrode where you can pass through a lot of electrical current, which will cause excitotoxicity in a brain area and burn it out. Or you can inject certain chemicals like acids, which will burn a hole and destroy the neurons in a particular brain area. So this has been done for all of the song system nuclei within the song control system and this has been one way to prove that it's involved with song learning and specifically. This is an example of experiments that were done by Fernando Nottebohm, who was one of the first people to do a ton of these kinds of experiments and describe the song control system in the 70s. But this is from a review of his published in 2004. This is what a canary song looks like. So they have a whole bunch of notes that they'll repeat over and over and over again, and then they switch, and then they do it again 10, 12 times, and then they switch, do it again. Very typical canary kind of song. Long, sweet, pretty. That's why people keep canaries. So Fernando had recorded this bird and got this nice song, and this was the day before he did a surgery where he anesthetized the bird and then burned out one of the HVCs. And then he let the bird recover. 6 days later, this is what the song looked like. You can see that it has some elements of it, but it's totally messed up. And he only burned HVC on one side of the brain. He left the other side intact. And it had this kind of impact on the song. And if you do it to the other side, you get a similar impact. If you do it to both sides, you can basically obliterate song. So they need both sides intact. Disrupting one side will really mess it up. So they're clearly using both sides. Okay, so that's one way of knowing how a brain area is involved. You destroy it. But it's a pretty abrupt way of testing the role of a brain area involved with a particular behavior. We knew that these song system areas are involved in song because we went in and destroyed them and observed their effects on behavior. We've done it in RA and the other brain areas, and we'll talk about that in a little bit. But more sophisticated techniques came about in the late 90s and early 2000s where we could record neural activity in awake zebra finches. So this involved using a glass pipette that could be attached to a motor drive, which then has some electronics attached to it, connected up to an amplifier so that you could record brain activity in a live awake zebrafinch. So this is an example of what this looks like. So this animal has this motor drive, and it already has an electrode that's been placed into the brain. And because there's the motor drive there, you can record from a neuron and observe the activity over time. But then you can drive the motor and make the pipette go down a little bit farther and record from a different neuron in the same animal and observe how that neuron fires. And then move the motor again and then record from a different neuron. And you can record from many, many neurons from one animal over a period of hours or days using this technique. So it's a really powerful technique. And then we can observe what's happening in these brain areas over time. Remember, so we're going to talk about some raster plots. And remember, raster plots will show change in activity over time across many trials. So this is what this would look like with this kind of in vivo recording technique. This is some example of where we have given 20 trials to an animal. Then at time 0, something happened. Say, in the example I've used in the past, I said that like light was shining on this. And then we see that there's an increase in activity. It's not stereotyped, though. There's not any sort of clear pattern here. You can see that from trial to trial, every single trial, there are more spikes post-stimulus than pre-stimulus, right? There's many fewer here than here. That is obvious. And that's the advantage of a raster plot. Sometimes you can see patterns of activity, though. This is not the case in this raster plot, but you might see some sort of major increase, maybe 200 milliseconds afterwards, and then it might go down, and you would be able to ascertain that. What does this look like in songbirds? And so songbirds, that's what we're going to see over the next several slides. With songbirds, we have incredible precision of activity. So this is an example of activity when a bird was singing a particular motif. This bird had only 4 notes in his motif, A, B, C, D. And these are different neurons that were recorded. And he in this study, they recorded 8 different neurons, and they recorded them 10 times. whenever the bird was singing. So what you can see is that in HVC, if you're recording from one single neuron, this neuron basically fired once. It fired a couple of times, but within the space of about 5 to 10 milliseconds. And it always fired at the same exact time within the song. Then if you recorded from a different neuron, now you have a neuron that fires at a different time within the song, but again with the same precision. If you look at a different neuron, a different place. Another neuron, a different place in the song, but again, very, very similar precision. You find some neurons that will fire at other times within the song with a little bit less precision, but you can see that it's very specific to a part of the song. And then here's another neuron that fires at this part of the song. Essentially what's happening is that you're only looking at 8 different neurons here, but because HVC has hundreds of thousands of neurons in it, you could imagine the sum activity of those neurons is to allow for the precise motor output, and that individual neurons will have a note that they have to play at a particular time. It's almost as if there's an orchestra, and you're going to have the song being produced out of the orchestra, but each musician within the orchestra has one note that they have to play, and that they will fire, and then that's their job for that particular part. And this is very typical for HVC, that we see just individual firing, and then this is essentially led to a lot of our understanding of how motor control is happening, and that there must be some sort of computational process by which this occurs. There's a ton of interesting stuff happening with the electrophysiology of these brain areas. This has also been done in RA. So neural activity in RA is still incredibly precise, but individual neurons there are firing multiple times within a song. So they don't fire just once, but they fire multiple times. And that sort of makes sense. It's because we have HVC that projects to RA. What's happening is that the RA is summing some of the activity within HVC. So each individual RA neuron must be getting multiple inputs from multiple HVC neurons, and it's the summation of that that's now allowing for a little bit more firing happening out of RA. Then that information is transported down to the tracheal syringeal portion of the hypoglossal nucleus, or N12, which then coordinates the activity of those motor units that are synapsing onto the syrnix to allow the muscles to contract in a very precise way. So that's a lot of what we know about the descending motor pathway. It is specifically involved in song production. If you lesion these brain areas in either juveniles or adults, it will lead to the abolishment of song, and that neural activity in HVC appears to coordinate neural activity in RA, and neural activity in RA, each individual neuron might fire multiple times, but each individual neuron is firing at different times, but all of that is being summed here in this portion of the N12th nucleus, and then that is going to lead precise coordination of motor output. What about this red area? This is known as the anterior forebrain pathway. You can see that it has a projection from HVC. There's some neurons here in HVC that project to this area, which is called Area X. Area X projects to this portion of the thalamus called DLM. DLM projects up to LMAN, and then LMAN projects down here into this essentially motor cortex area, RA. This should look familiar to you. In addition, we have portions of the substantia nigra pars compacta, the dopamine neurons, projecting up to Area X. This looks very much like a cortical basal ganglia thalamocortical loop, where then we have an output that is going to be regulating the activity outside of RA. So that's essentially what the anterior forebrain pathway is, and we're going to walk through how this pathway works a little bit. Interestingly, lesions of Area X have little effect on adult song production. So these are examples of 2 different birds. So this is the day before pre-lesion, and then lesioning 16 days after. And then this is a different bird. Its motif looked like this. 1 day pre-lesion, 45 days after, it still looked pretty much the same. It turns out that if you lesion area X in adults, it doesn't have a whole lot of effect on the song. It's not perfect, and it does change over time, and it gets worse and worse over time. But it turns out that you don't necessarily have to have the basal ganglia intact in order to produce an already established motor output, and have that output be affected too much. It will be changed over time, and it will degrade over time, but the establishment and the basic production of it isn't utterly dependent on having an intact basal ganglia. What is important, though, is during the learning process, and that's why the basal ganglia really is this learning pathway. And here's an example of what this looks like. So there are a few ways of kind of analyzing the effects of lesions and changes in song. One is just to look at the consistency. And so instead of using letters here, this is a student of Fernando Notebaum's lab. Her name is Constance Scharf. I actually worked in her lab for 3 years. She's a professor of neuroscience in Germany. And so what she had done is analyzed the consistency of song. Instead of using letters, she used numbers here. So as we said, zebra finches have these introductory notes, and then they will go to A, in this case 1, and then they go to 2, 3, 4, 5, 6, 7. Sometimes the bird will end there, or it'll continue and add another note called 8, and then 9, and then go to 1, and then go around and around and around, and then it always ends at 7. That's just what this bird does. It's the song that it learned, and it'll do it like 3 or 4 times. And you can look at what the motif consistency looks like. 1 is always preceding 2, which precedes 3, which precedes 4. There's no variability. If you lesion area X between days 30 and 46, this is what the song will look like. They will learn some of these elements. They learn that there's like 1, 2, 3, 4, 5, 6, 7, and 8, but there's no consistency in it. The pattern of the motor output is severely disrupted. With this bird, it did happen to always, once it sang 1, it always sang 2, but after that, it would sometimes sing 3, but sometimes it would sing 6. 3 would sometimes sing 4, but also sometimes sing 5, more usually because it's a thicker arrow here. You can see with 8, after 8, who knows what's going to happen? It can sing 2 or 3 or 4, sometimes go to 7. 7 can go to 8 and then to 7. It's all messed up. Tons of extra arrows here. There's no consistency. So the pattern of motor output is severely disrupted when you lesion during the learning phase. And so that can't inform the cortex on how the pattern is supposed to really look like. Here's a nice way of illustrating this effect and what happens as far as the effects of lesions of HVC versus the what happens for intact birds. So you can give a score to these things. So the consistency and linearity, so how often they are able to sing the song in the right order, and then how consistent they are from motif to motif. So every time that they sing it, we can give a score. Birds that are intact, that's the empty circles here. You can see they have a high linearity score and they have a high consistency score. But if you have birds that have lesioned area X during this learning phase, you can see this entire group has a lower consistency score than these guys and a lower linearity score than these guys, except for a couple of examples. But you can see the majority of them are now singing total garbage. Their consistency and linearity is severely disrupted. All right, so some of you might be thinking like, okay, well, how does this relate to our understanding of the basal ganglia in mammals? These kinds of experiments haven't been done extensively in mammals. So we actually just know more about this from songbirds than we do in mammals, but there have been some experiments. So here's an example of an experiment that was done illustrating how this works this was done in monkeys. And so they had trained these monkeys to do a sequence of actions. It was like some lever pulls depending upon like visual stimulus, and they had to do it in the right order. And then they lesioned different parts of their brain. They lesioned 3 different areas. They lesioned the amygdala, which we'll talk about, but it's more related to fear and also emotion. They lesioned the medial striatum, MS. They also lesioned the orbital frontal cortex, and we're going to talk about that as well and its association with reasoning. Interestingly, When they lesioned the striatum and the orbital frontal cortex, they found that if the monkey made an incorrect response, that it had a much harder time adjusting its behavior. So the orbital frontal cortex is heavily involved in reasoning and logic. And so if you lesion that area is not working properly. But also the striatum, basically they have already learned this pattern, and once you lesion it, they're unable to make adjustments in their shifting after an incorrect response. The probability of shifting after a correct response, so this would be like, all right, you're not supposed to change. This is where they see an interesting difference. So because they've lesioned the orbital frontal cortex, they're just making more mistakes. They are shifting after a correct response when they really shouldn't. But this is not true with the striatal lesions. With the striatal lesions, again, they're still being very consistent with the motor pattern that they're making. They're unable to recognize when they make mistakes. But they're also still very good at keeping the pattern intact, even with the striatum completely lesioned. They lesion the amygdala here kind of as a control. So you will always want, when you're doing these kind of lesion studies and you just mess up behavior, you want to see, okay, well, what's the likelihood that it's just because of the surgery? So you lesion in unrelated areas. So in this case, these animals had a lesion amygdala, and they were still able to adjust and make changes after they make an incorrect response. But with the medium striatum lesions, they're still very consistent. They are not able to make those adjustments. So in this kind of experiment and other experiments with the songbird has also shown that essentially the basal ganglia acts as a training tutor tool for programming the motor cortex so that it can make the correct pattern over time. But and then it also acts as an error detection mechanism. And this appears to be a major role of the basal ganglia. Again, we actually don't have that many experiments done with mammals in order to really test this, though. So one thing that's interesting about the anterior forebrain pathway is that we have talked about how there is the cortical basal ganglia thalamal cortical loop. Remember, though, in mammals, basal ganglia includes striatum projecting to globus pallidus, right? That we don't necessarily see distinct units like that. Instead, we have this one area that makes up the song control system called Area X, receiving cortical input, and then that same area projecting to the thalamus. It's almost as if that this area is somehow we're skipping one of those steps. That seems really weird. We do know that it gets dopaminergic input, so it does seem to have a lot of the characteristics of basal ganglia. But what about the striatum and globus pallidus parts? Clearly, that's an important way that this works, right? So let's talk a little bit about that. A series of experiments have shown that this is a rendition, a very simplified and very nice schematic of the mammalian basal ganglia pathway, with the cerebral cortex have an excitatory input onto medium spiny neurons in the striatum. Those medium spiny neurons inhibit these tonically active globus pallidus neurons, and then those normally will inhibit the thalamus, but when they are inhibited by the striatum, then they are disinhibited, the brake is removed, and the thalamus can fire. That is how we understand the mammalian basal ganglia. But when we talk about the song control system, it superficially looks like it doesn't exactly match. We have HVC projecting to Area X, which then projects to DLM. It looks like that there is a step missing. Instead, a series of experiments done by David Perkel and people in his lab, they have shown that HVC, which is cortical in nature, projects to medium spiny neurons in Area X. And then those medium spiny neurons, they don't project out of Area X. Instead, they synapse onto these very large projection neurons that are within Area X that are tonically active and inhibitory, and then it's those neurons that synapse onto DLM. So essentially what happens, what this means is that Area X is a blending of striatum and globus pallidus. It's not distinct like it is in mammals. Instead, what we find in birds and actually lizards and frogs, it turns out as well, that the globus pallidus is mixed, that the basal ganglia, you have medium spiny neurons, they're right up next to these globus pallidus-like neurons. that are tonically active and inhibit the thalamus. So this is essentially the activity that you're going to see, and you'll see that it matches what we know happens through the basal ganglia pathway. Cortical area, it gets a signal saying, hey, I need to sing. So HVC starts to sing, and HVC starts to fire, I should say. There's a medium spiny neurons also then become active, and then they inhibit the projection neurons, which disinhibits the thalamus so the thalamus can fire and then allow for the production of song. So you can see how similar it is, despite the fact that it's blended and mixed together in one nucleus called Area X. There's a bunch of different experiments that have been done with the role of dopamine. Ton of really interesting experiments, too much for me to get into. But here's one just basic interesting experiment. So I've talked about how song is used to attract females. And zebra finches, they will adjust their song a little bit, depending upon whether they are singing to a female or not. So they do what's called directed song. So if there's a female present, that male will direct his song to the female. He'll be paying attention to the female. He's trying to court that female. There's also undirected song. And what we find is that under the directed song conditions, when they're singing to a female, the song tends to be more consistent. It's less variable. When the male is also singing towards a female, we find that there's also an increase in the amount of dopamine that's there. So in this experiment, they had tested these males that had a tube placed in the area X. And with this little tube, they were able to sip out tiny little bits of neurotransmitter. And what they were measuring is the amount of dopamine. So they can measure the precise amounts of dopamine that are coming into the area X at any one time. This is actually very similar to the kinds of techniques that are used in the Buzinski lab here in the School of Neuroscience. He does this similar kind of microdialysis type technique, also looking at dopamine as well as other things. So in this case, what we see is that when these males are singing towards a female, they have a higher amount of dopamine that's present when compared to when that same bird was singing in an undirected condition. So when there's no female around, there's less dopamine present than when there is a female around. So they all have the surge of dopamine. They're using it to attract a female. It's a sexually exciting type of behavior. And boom, sure enough, there's an increase in dopamine, which is probably why the song tends to be more consistent and stereotyped, because you have more dopamine there to reinforce and promote better activity through the basal ganglia. We will be discussing the influence of a particular gene called FOXP2 over area X and how it regulates song. But I want to take a little bit of a segue and talk about the field of neurogenetics. Neurogenetics is a very important field. We're not going to discuss it enough in this class, but it's essentially the study of how genes and gene mutations lead to affect behavior, mental illness, and overall brain development and function. One of the most famous genes that have been discovered in the field of neurogenetics is FOXP2. FOXP2 is a transcription factor that is expressed in the striatum. We'll talk about what that means, transcription factor, in the next couple of slides. But it's essentially a factor that regulates cell growth, proliferation, and differentiation. Its significance was initially identified in the KE family. So this is a family that's in the United Kingdom. Simon Fisher had heard about this family, that they have a very specific speech and language deficit, and that they said they have a very difficult time learning language and producing language, primarily the production side. So the black marks here this is their family tree. So multiple generations, and the black marks here indicate affected family members. Each of these family members was carrying a mutation for FOXP2. And so you can see that it's across multiple generations where you have affected family members, and the affected family members only have the mutation. The unaffected family members do not have the mutation. So this is an autosomal dominant speech disorder. So by inheriting the mutated gene, they had these mutations. FOXP2 is the major focus of the Bowers lab that's in the School of Neuroscience. He studies how FOXP2 and other FOXP members control vocalizations in rats, with major focus on sex differences in these different FOXP members and its contribution to the neural mechanisms of vocalizations in rats. So what does FOXP2 do? I mentioned it's a transcription factor. Obviously, there's a gene in there, so we have our DNA, and that there are these promoter regions which will drive the expression of FOXP2. So at some point, FOXP2, the gene will get turned on. We'll have some sort of transcription factor that will bind to the promoter region, leading to the production of FOXP2 mRNA, right? So the gene makes mRNA, and then the mRNA has to be transcribed and converted into protein. So this is what the FOXP2 protein looks like. And now we have this protein around. So what does that mean? So what does the FOXP2 protein do? Well, I mentioned it's a transcription factor. So it means that this will bind to FOXP2 promoters, which will be ahead of certain target genes. FOXP2 tends to act as a transcriptional activator. So it will bind to transcription factors such as this and this that will allow and then turn on the expression of certain genes. But there are also FOXP2 promoters that will suppress the activity of other genes. So when FOXP2 is turned on, you're going to have changes in expression of all kinds of other genes, such as related to differentiation, cell proliferation, and also dendritic arborization. So how do we identify cells that are expressing FOXP2 protein? We use what is called immunohistochemistry. You had learned about immunohistochemistry in the laboratory portion for Neuro 1, but I'm going to walk through the basics of immunohistochemistry here. So this is the transcription and translation portions, right? We got the mRNA and we have the protein. There's lots of protein that gets expressed if this gene is turned on. And in the cell, then we can have antibodies that will bind to these proteins, and then we can do immunostaining that'll allow us to see what those cells look like. So I went ahead and did this in zebra finches that were killed at 35 days after hatch. So they were only 35 days old during that phase of learning. Zebra finches that were 50 days old, and then we had birds that were adults. These birds happen to be singing, and that will be relevant in a few more slides. So we had sections that were stained with Crescel violet. So this is the Nissl stain sectioning, and you can see area X is located here. It's kind of a lamb chop shape. And then the rest of this is basal ganglia, and then this would be basal ganglia circuits that are not related to singing, doing other motor-related functions. But this is going to be the song circuit here, area X here. And then I had an adjacent section, so this would be section A, and then this is section B, and I stained these for immunohistochemistry for FOXP2. And the darker it is, the more FOXP2 positive cells are, and also how intense the cells are. And what you can see is that there's a ton of FOXP2 in this striatal basal ganglia portion of the brain. So we're looking at the striatum here, and there's tons of cells here, and it's particularly intense inside area X. This was true at day 35 as well as day 50, but something very interesting happens in birds that are adults. You can see that there's still plenty of FOXP2 expressed in the striatum outside of area X, but inside area X, there's hardly anything. You can see this is the border of area X. There's very little FOXP2 that's expressed there. But on the outside of area X, there still is quite a bit of FOXP2. This is what it looks like on the border in juvenile zebra finches. You can see there's plenty of FOXP2 positive cells inside and outside. When we looked at the staining, we noticed that there were two different types of FOXP2 stained cells. We saw cells that were really intensely stained, and so that's what these arrows are pointing at. These are very, very dark cells. But then we also saw a lot of faint, weakly stained cells. So we have intensely stained cells and weakly stained cells. And there's quite a few of them. There's a lot inside and outside area X, a lot of intensely stained cells and weakly stained cells. This is true at day 50 and day 35. But when we looked in the singing birds, really all we saw for the most part was weakly stained cells, and not a whole lot of them, especially in singing birds. We did see some intensely stained cells, but there really weren't that many, and not nearly as many as there were in the juvenile birds. So what's going on here? Why are there these differences in these different kinds of cells? Well, the first thing is to quantify these things. So we looked at the density of intense FOXP2 immunoreactive cells, And you can see that the numbers of intense FOXP2 cells is much higher in the juvenile birds than it is in the adults. So adults have some, but not nearly as much as in juveniles. In fact, it's about a 10-fold decrease. Now, for the weakly stained cells, in fact, it turns out that there's no significant difference across the different groups of the number of weakly stained cells. And if anything, adults have a few more of these weakly stained cells than juveniles. So it looks like there's this flip. A lot of them in young ones, a lot of these intense in young ones, and then basically no change, or if anything, maybe an increase in the number of weak FOXP2 positive cells. So this, one thing that we know about developing brains in these juveniles is that they still have neurogenesis going on. So there are new neurons that are being born and incorporated into these brain circuits. So we did an experiment where we injected birds with what's called bromodioxyuridine, or BRDU. BRDU is a version of thymidine that gets incorporated into newly made DNA. So when cells are undergoing DNA synthesis and there's chromosome duplication happening, this is an example of the cell cycle here. So during the S phase, or synthesis phase, we have new DNA being made, and if there's BRDU around, then that will substitute for the thymidine in the newly made DNA. And then you can use immunostaining to label those cells days, weeks, months, even years later to identify which cells were dividing at that particular time. And then we can identify those cells that were born on that particular day when the animal was injected with BRDU. So it's a way of tracking how much neurogenesis was happening on a particular day. And we can also look at the survival over time. And so those cells will be labeled. So we did this with zebra finches injected at various time points. We injected birds on day 79 in this experiment. So these were still relatively young birds. So they were injected at day 79, and then we killed them 21 days later. So this was 3 weeks later. And then we did staining for FOXP2 and BRDU. And what we found is that the majority of the BRDU positive cells were intensely stained. So these were the very bright, in this case, now we switched to fluorescence instead of using the colorimetric staining that we had before. But the green is FOXP2. You can see that there's intense ones and there are weak ones. And in general, what we found is that many of the BRDU positive ones were these intensely stained FOXP2 positive neurons, whereas the weakly stained ones, there were some, but they're not nearly as many. You can see that in this field, there's around 50 or so weak stained ones, and there's only one BRDU positive cell out of around 50. And then in this particular slide, there's 5 intensely stained ones, and 2 of the 5 are intensely stained. So we went through and quantified this, and we found that the percent of FOXP2 positive neurons in these relatively young birds that were intensely stained was much higher than they were in the weakly stained ones. What this indicates, so these neurons were born 21 days before, and that they were likely to be newborn neurons. So in one of these animals, nearly over 40% of all the FOXP2 aminoreactive neurons were intensely stained versus something like 2% in the weakly stained ones. So what this is indicating is that newborn neurons, and in young animals you're going to have a lot more newborn neurons, are going to be intensely stained for FOXP2, and then as they mature, they lose that intensity, and then they become weakly stained. So that seems to be the developmental change. So what role could this be? Like, how might FOXP2 be important for then the learning part? If there is this, we see this age-related decline in FOXP2 expression, and that it appears to be related to neurogenesis, which is going to be really important for how these circuits get established. So one way to test this is to do short hairpin knockdown of RNA for FOXP2. So what happens here is that we can design a virus that will express a kind of alternative RNA that will knock down the FOXP2 here. So it'll bind to each of the little FOXP2 mRNAs, which will eliminate those, which means that there will be no protein. So the protein will disappear, and then we can knock down the expression. So this is one way of manipulating the expression of FOXP2 in live animals. And then this expression of the virus that'll be making these short hairpins for weeks. So this is an experiment that was done also in the Scharf lab. The Sebastian Heisler, he had injected lentivirus into area X on both sides of the brain at birds that were 20 days old. So these are quite young birds. It's just as they're in the sensory phase. And so they're going to have FOXP2 that's going to be knocked down during the sensory and sensory motor phase. And then he compared what the short hairpin looked like for the animals that had FOXP2 knocked down in their similarity to the tutor song that they were exposed to. And you can see the birds that had short hairpin FOXP2 expression in area X had very poor similarity scores, 60% similarity, compared to birds that were injected with virus and then were expressing a short hairpin that was different than FOXP2. It was a control virus. And that these birds were basically no different than control. A typical zebra finch will copy about 90%. They'll usually have a 90% similarity score. But if you mess with FOXP2, you reduce it significantly. So clearly, FOXP2 seems to be playing an important role during the song learning phase as well. So this is an indication that FOXP2 is involved with the establishment of these brain circuits that are really critical for learned vocalization. It's likely this is true. This is clearly true in zebra finches, but something similar is probably happening in humans as well. So this is essentially a summary of what we've seen with FOXP2 expression over age and behavioral context. We know that FOXP2 is expressed in the proliferative zone from experiments that I didn't really show, and that they're expressed in migrating neurons. Again, this is some data that I had collected that I just don't have time to show. So they're found in the proliferative zone. They're found in migrating neurons that are migrating through the tissue. It takes about a week or so for them to get into area X. And expression is really, really high in these young, still establishing themselves baby neurons. And then once they differentiate and they turn into neurons, these are medium spiny neurons. So this is what the dendrites look like. They're kind of spiny. Then the expression of FOXP2, which is a transcription factor located in the nucleus, its expression goes down. See, it's weak. This is the weakly stained neurons. This is the intensely stained neurons. Later on, what we find out is that it turns out that singing or not singing will affect the expression of FOXP2. So it's probably leading to some subtle regulation of changes in FOXP2 neurons over time. That there's some behavioral differences that happen between non-singing and singing birds that is too much for me to get into. Essentially, though, the take-home point is that FOXP2 expression is really high in very young neurons, and then its expression goes down. And that this high expression is probably critical for how these neurons differentiate and establish themselves in the circuit within area X. And so how the connection between HVC connects to these neurons, as well as how these neurons connect to the palatal neurons that are the projections to the thalamus. When you throw in that virus, it interferes with this so that these neurons aren't able to express FOXP2. And so they're going to have a misalignment in how they establish themselves within the neural circuit. So that's the summary. Here's some key questions for this lecture. What are the key experiments that show that song is learned? What are the similarities and differences between birdsong and human speech? For what purposes do birds sing? How do we know that a brain area contributes to song production? What is the basic role of HVC and Area X in song memorization and production? What are the ways that the anterior forebrain pathway is similar and different from the cortical basal ganglia thalamal cortical loop in mammals? And then also, how does FOXP2 expression change over a cell's lifespan? And what does this suggest about FOXP2 expression does for medium spiny neurons, how they get incorporated into neural circuits? So that's it. It's a lot to cover, I understand, but it's a super interesting system. I hope that you appreciate it, and we'll see you next time.